My name is Shelley Arnott, and I'm a professor in the Department of Biology at Queen's University. And my research group studies aquatic ecology, and more specifically, we look at how aquatic systems respond to environmental change. Canada is rich in aquatic resources. In fact, 20% of the world's freshwater is located in Canada. And you can see by this graph that the blue area shows regions of the world where more than 10% of the landscape is covered in water. And Canada has a lot of that dark blue color, primarily in the boreal shield, but a good indication of how much water we have. And this water provides a number of important services. Things like drinking water, irrigation, and recreation, as well as generating hydroelectricity, are all important services that we get from water. Despite the importance of freshwater in terms of services and culture, our freshwater systems are facing many challenges. And this includes things like increased nutrient loading, invasive species, pollution, climate change, and habitat alteration. We use a number of different ways to study how aquatic systems are influenced by these challenges. And this summer at QUBS, we're employing mesocosm experiments to determine how salinity, nutrients, and turbidity influence aquatic ecosystems, and in particular the zooplankton communities. Now, zooplankton are a central component of aquatic food webs. They graze algae, so they transfer energy from primary producers up through the food chain to higher trophic levels, such as fish. One of the environmental challenges that we've been interested in addressing is the influence of road salt on aquatic ecosystems. And in the winter, to increase safety on roads and sidewalks, Canadians apply up to 7 million tons of salt every year. And this salt eventually finds its way into aquatic systems. And what we've noticed is that the concentration of salt, or sodium chloride, is increasing in many lakes, especially those that are located near roadways and urban areas. So this is the mesocosm field at QUBS. So we have rows of these 200-liter tanks that we've filled with water, and we can manipulate the zooplankton and the conditions, the environmental conditions of the water. And so in Carrie's case, she's manipulated turbidity and nutrients, and Kaylin has manipulated calcium and sodium chloride, which is to represent road salt. Okay, so Kaylin, can you tell me what you're doing?
Sure. I am rinsing an 80-micron zooplankton net right now so that I can prepare to take some zooplankton tows from this lake. This is South Otter. We are towing for zooplankton to stop my mesocosm experiment, which has 60 mesocosms that are filled to 180 liters. So we need to take as many tows as we can to match that total volume of water. That is what we're working on today.
So what question are you asking with your mesocosms?
I am investigating interactions between chloride and calcium influences on zooplankton community structure. I am investigating, we project that at higher calcium levels we'll see ameliorated toxicity effects from chloride.
These containers are filled with zooplankton that we've collected from the lakes, and we're going to stock the mesocosms with them. So you can see these little dots swimming around, and those are different types of zooplankton.
Hi, I'm Michael. I'm a fourth-year biology student, and I'm a SWEP student in Dr. Arnott's lab.
And what are you doing this summer?
This summer I'm assisting with all the different mesocosm experiments that we have going on up at QUBS. Yeah.
Good. And Harina, do you want to introduce yourself?
Hi, my name is Harina. I'm a recipient of the QUBS Fellowship, and I'm working here at Dr. Arnott's lab for the summer.
And what are you doing?
I am also assisting in mesocosm experiments here at QUBS.
Carrie. I'm a master's student in the Arnott lab at Queen's, and I'm standing beside my mesocosms because this is my master's project. So I'm investigating how turbidity and nutrients affect aquatic communities, specifically zooplankton and also phytoplankton. This is actually a great example of how the turbidity of water can change. So this is a very turbid mesocosm. It's very cloudy. Turbid is another word for murky, while this is a much clearer one because we didn't add any sediment to it. So yeah, my project sort of stemmed from the issue of permafrost thawing in the Arctic and causing a lot of lakes to become very murky, which completely changes the biology, chemistry, and physics of the lake. But it's a problem found elsewhere in the world as well.
What causes turbidity?
It's always caused by something suspended in the water. Algae can cause turbidity, but more commonly it's caused by inorganic particles suspended in the water. So that could be anything from clay to like really, really fine particles of any type of dirt. Picture like a really fast moving river. Typically that picks up a lot of sediment and gets it suspended in the water, and it looks brown and muddy, kind of like that.
So would anything cause turbidity in lakes around the QUBS area? What kind of activities would increase turbidity?
Yes, construction can increase turbidity. The construction of roads, erosion in general can. Yeah.
So it's an issue not only in the high Arctic, but also around here.
Exactly.
Okay, so what are you doing here today?
So today we are collecting water to measure the total nitrogen concentration. So the primary focus is turbidity, but we're also interested in how nutrients interact with that stressor or variable. So we need to collect water in these, and there can't be a single bubble. It has to be completely full. And we'll send it away to a lab, and they're going to tell us how much nitrogen there is. Okay. We've tightly rolled our covers to keep any bugs out. This year specifically gypsy moth caterpillars. So you have to rinse the container 3 times. And then you want to get all the bubbles out, and you actually want to cap it underwater. So that there's no air.
Excellent.
Thank you.
Hi, I'm Eric Massa. I'm a PhD student in Dr. Shelley Arnott's lab. I am researching the effects of nutrients and salt on zooplankton and algal communities in the freshwater lakes of the QUBS area, and what the effects of heat are on that as well.
So why are you interested in salt and nutrients?
So I'm interested in salt and nutrients for a couple different reasons. One of the big ones is I'm from Florida, where saltwater intrusion into the bedrock is a big issue. And nutrients coming from agricultural headlands into our freshwater are also huge issues. Obviously, salt in the winter is a bigger issue in Canada rather than seawater intrusion, but similar effect on water concentrations of chloride.
And why are nutrients important?
So nutrients are used to, by the algae, they grow much more readily when they are not nutrient limited. So that's whether that's phosphorus or nitrogen. And seeing how the heat affects that, and thus how that filters up to the zooplankton community, who are independently affected by chloride and heat as well.
Why are you interested in heat?
So between global climate change, seasonal change, heat is a big influence on the environment.
So what's your big question?
Big question is what can we see from multiple stressors interacting at the same time on freshwater communities.
How are you addressing the interaction between chloride and heat and nutrients and heat?
Okay, so this is field season 1 of a 2-part experiment. So this field season is focused on what the independent effects of nutrients and chloride are, as well as the effects of nutrients and heat and chloride and heat. And then next field season will be looking at, okay, so we know that this combination of heat and nutrients has an effect, whereas without heat it does not. Same thing for chloride. Okay, what happens if we take this concentration of chloride and nutrients that are affected by heat, put them together? Does that have the same effect? Does it have a greater effect, a lesser effect? Do they cancel out? Probably not. Just going to go out on a limb and say that.
How are you testing these questions?
So I'm testing these hypotheses, questions, whatever, through the use of a mesocosm experiment. So I have a total of 48 mesocosms. The mesocosms are assigned treatments, so whether it is a heated treatment or not, and also assigned whether it is a salt treatment or a nutrient treatment. And then in the chloride and the nutrient treatments, they're randomly assigned on the gradient what they'll be.
Okay. Anything else you want to add?
QUBS is beautiful, and it's absolutely wonderful being out in the field, especially after the last year and change in the pandemic.
Yeah, I agree. Thanks.